In this lesson, we are going to discuss the molecular basis of inheritance. At the end of this video lesson, you should be able to describe the structure of nucleic acids and differentiate between DNA and RNA. The chromosomal theory of inheritance notes that genes are found on chromosomes and that the action of chromosomes during meiosis accounts for the loss of segregation and independent assortment of Gregor Mendel. However, the chromosome is just a bigger picture of what inheritance looks like. Each chromosome is made of protein and a nucleic acid. Nucleic acids are polymers of nucleotides that store, transmit, and help express hereditary information. These nucleic acids enable living organisms to reproduce their complex components from one generation to the next. Each long chain of nucleic acid is made up of a series of nucleotide units. A nucleotide has three major components: a phosphate group, a pentose sugar, and a nitrogenous base. A pentose is a monosaccharide or simple sugar with 5 carbon atoms. Pentose sugars in nucleic acids are present as closed-chain or cyclic sugars. The position of the carbon atoms in the sugar is very important to determine the orientation of the nucleic acid. Each corner or edge in the sugar is a carbon. Carbon 1 is on the rightmost carbon of the sugar. Carbon 2 is on the lower left portion of carbon 1. Carbon 3 is on the left of carbon 2. Carbon 4 is on the upper left of carbon 3. And carbon 5 is positioned above carbon 4. There are 2 pentoses found in nucleic acids, ribose and deoxyribose. The 2 pentoses are the same for all carbon atoms except for carbon 2. For ribose, a hydroxyl group is present below carbon 2. For deoxyribose, the hydroxyl group is deoxygenated, thus the name deoxyribose. Now, let us discuss the nitrogenous bases. These are called nitrogenous bases because nitrogen atoms appear to take hydrogen out of the solution, serving as bases. There are 2 nitrogenous bases families, purines and pyrimidines. Purines are bigger, with a 6-sided ring incorporated into a 5-sided ring. The purines are adenine and guanine. On the other hand, there is one 6-sided ring with carbon and nitrogen atoms in pyrimidines. Cytosine, thymine, and uracil are members of the pyrimidine family. The specific pyrimidines and purines differ in the chemical groups attached to the rings. These nitrogenous bases form base pairs which are 2 chemical bases bonded to one another. Adenine forms a double bond with thymine. It may also form a double bond with uracil. Lastly, guanine forms a triple bond with cytosine. These bonds in between the nitrogenous bases are weak hydrogen bonds. Another important information about these base pairs is that these are present in all organisms. According to Erwin Chargaff, DNA composition is species-specific. This means that the percentage of the nitrogenous bases varies per species. These percentages usually denote what species an organism is, or how related is a certain species to another species. Since this is universal, Chargaff came up with a rule named after him. Chargaff's rule states that the amount of adenine is always almost equal to the amount of thymine, and the amount of guanine is always almost equal to the amount of cytosine. To understand this, let us compare the nitrogenous base compositions for 4 species: humans, ox, wheat, and yeast. We are going to compare the adenine to guanine ratio, thymine to cytosine ratio, adenine to thymine ratio, cytosine to guanine ratio, and lastly, pyrimidine to purine ratio. These data show that for all species, the ratio of the nitrogenous base pairs belonging to the same class is not equal to 1, not even almost equal to 1. But when looking at adenine to thymine, which is a purine to pyrimidine pair, and cytosine to guanine, which is pyrimidine to purine pair, the ratio is 1 or almost equal to 1, having deviation not greater than 0.1. This means that the pyrimidine will only pair with the purine and vice versa, as indicated by the ratio of pyrimidine to purine in all species, This validates the rule that adenine pairs with thymine and cytosine pairs with guanine. Let us look at one example to practice Chargaff's rule. A DNA sample yielded 24% adenine. What is the percentage of the other nitrogenous bases? We are asked to solve for the percentage of thymine, cytosine, and guanine, given that adenine makes up 24% of the DNA. What we know is that adenine pairs with thymine, and that the percentage of adenine should be equal to the percentage of thymine. That means that thymine is also 24%. This gives us 48% of adenine-thymine base pairs. To know the percentage of cytosine and guanine, we must first get the total percentage of the cytosine-guanine base pairs. We should subtract 48% from 100. This gives us 52%. To get the individual percentages of cytosine and guanine, we need to divide the total cytosine-guanine percentage to 2. Thus, cytosine and guanine each comprise 26% of the DNA. Now that we already know the components of a nucleotide, we can now build the entire nucleotide building block of nucleic acids. Phosphate is connected to the pentose sugar, but now without the hydroxyl group on the 5th carbon due to a dehydration reaction. This creates an ester bond between the two. On the right side of the pentose sugar, the nitrogenous base is connected to the 1st carbon, also removing the hydroxyl group to create a glycosidic bond. The pentose and nitrogenous base forms the nucleoside. Now that we have the building block of a nucleic acid, let us now discuss the two important nucleic acids, the DNA and RNA. The DNA or deoxyribonucleic acid is the nucleic acid mainly responsible for the storage of hereditary information. It is named as such because the sugar in this nucleic acid is deoxyribose. Aside from the sugar component, an important feature of the DNA is the double-strandedness, which is formed by the base pairing of adenine and thymine, and guanine and cytosine. On the other hand, the RNA or the ribonucleic acid is another nucleic acid which functions in various gene expression events such as transcription and translation of the hereditary information. It is named as such because its pentose component is the ribose sugar. In comparison to the DNA, the RNA is a single-stranded nucleic acid. However, it still has base pairings, especially when doing its functions during gene expression. The difference is that instead of containing thymine, it has uracil. Thus, the base pairs in RNA are adenine is to uracil and guanine is to cytosine. These two nucleic acids are not simply complete or half ladders of nucleotides. These are actually helical or spiral. In 1953, American James Watson and English Francis Crick discovered the double helix structure of DNA. The brief but celebrated collaboration that solved the mystery of the DNA structure started shortly after Watson traveled to Cambridge University, where Crick studied the protein structure using a method called X-ray crystallography. Watson saw an X-ray crystallography photograph in the laboratory of Maurice Wilkins. As someone who is knowledgeable with X-ray structures, the two decoded that the structure of the DNA is a double helix. They were awarded with the Nobel Peace Prize for their discovery of the double helix structure of the DNA. However, it gained controversies because the owner of the photograph was actually not heavily credited. Rosalind Franklin, a highly experienced X-ray crystallographer, performed crucial experiments culminating in a photograph that enabled Watson and Crick to deduce the double helical structure of DNA. Images created by X-ray crystallography are not necessarily photographs of molecules. The spots and smudges were created by X-rays that were diffracted or deflected as they moved through aligned DNA purified fibers. The pattern in this photo implied that the helix was made up of two strands. With the extra information from Franklin and Chargaff's rule of base equivalences, Watson and Crick arrived at a model of the double helix. It also showcased the information, often an unpublished annual report of Franklin, that the sugar-phosphate backbones were on the outside and the nitrogenous base pairs are inside of the DNA molecule as shown in this figure. In the double helix of the DNA, we can see pairs of nucleotide connected by weak hydrogen bonds in the middle. Ladders of nucleotides are made possible by the phosphodiester bonds that connect the third carbon or 3' carbon to the phosphate group. This means that when looking at the molecular level, the 5' end of the sugar is nearest to the phosphate end of the DNA. The other side of the nucleic acid has an exposed hydroxyl group, giving us the 3' end because it has the third carbon that is on the outermost portion of the opposite side. This occurs both horizontally and vertically. But when considering a single strand of the double helix, the left side is parallel to the right side but is of opposite direction. The 5' end of one strand is adjacent to the 3' end of the other strand. Or in other words, for one strand, it starts with the 5' end and ends with the 3' end. For the other strand, it starts with the 3' end and ends with the 5' end. This gives the DNA an anti-parallel orientation. Thus, when writing a DNA strand, we will usually encounter a 5' end and the 3' end on both sides of the DNA strand. For example, we have a DNA strand with the first strand reading as 5', ATGCACGAT TAA 3', and the other strand being the complement of the first strand as indicated by Chargaff's rule should be 3' TAC GTG CTA ATT 5'. The strand which runs from 5' to 3' direction is the coding strand, and the strand which runs from the 3' to 5' direction is the template strand. To conclude this lesson, let us review the following key points. Nucleic acids are polymers of nucleotides that are made of a phosphate group, a pentose sugar, and a nitrogenous base. The bonds present in a nucleic acid are ester bond, glycosidic linkage, and hydrogen bond. Nitrogenous bases are classified as single-ring pyrimidines or double-ring purines. Chargaff's rule states that adenine bonds with thymine and cytosine bonds with guanine. The DNA is a double helical and antiparallel nucleic acid that is used as a storage of genetic information. And lastly, RNA is a single-stranded nucleic acid that has various functions during gene expression. It contains uracil instead of thymine. And that ends our discussion on the molecular basis of inheritance.